So today we'll study about different forms of cultures which are used and their applications in plant biotechnology, and what are the nutritional requirements of plant cells before starting with the different forms of in vitro cultures. So then, starting with culture initiation, we all now know that any part of the plant can be used as an explant to initiate different forms of cultures. So these plant parts are used to initiate and establish in vitro grown cultures. So why do you think in vitro plant cell culture or tissue culture can be of use? Because they can act as model systems to study the effect of various environmental parameters on the plant physiology and its growth and development. So because under in vitro conditions, which are controlled environmental conditions, you can study each factor at a time, while in whole plants under natural conditions, these the soil conditions or your environmental parameters are not under control. So field grown plants, we know that they can undergo photosynthesis, so you do not have to provide carbon sources or nutrients to these plants exogenously. They can themselves procure from the soil and use sunlight to produce the food source. But in case of in vitro cultures, this photosynthetic capacity is impaired. They do not carry out as large amount of photosynthesis as the whole plant does. So they need to be supplemented with exogenous carbon source, which is generally sucrose used as carbon source. Now what all different parts of the plant which can be used as explants? They can be nodal, they can be internodal segments, they can be apical or axillary meristematic regions, petioles, even your petals, flower petals, ovules, ovaries. So any part of the plant can be used effectively as an explant. Now how to decide which kind of an explant to be used? It also depends on what kind of in vitro culture you would like to carry out. So we will see when we will be discussing different forms of in vitro cultures, what are the explants used which can lead to different forms of in vitro cultures. So what should be taken care of when you are starting to initiate these in vitro cultures? If suppose you are bringing an outside plant material to inside to be used as an explant, first thing is you need to make sure that you bring it in an ice box or very quickly, so that the catabolic activity, as soon as you excise it from the plant, the breakdown begins. So you need to minimize that catabolic activity. So in order to minimize, you should bring it in an ice box and as quickly as possible. Now the second thing is, there will be contaminants if you are dependent on the outside plants as explants. So then you need to do surface sterilization, which is one of the key and the initial steps in initiating any kind of in vitro cultures. So how to carry out surface sterilization? When I say surface sterilization, which means that removing the surface contaminants. So first thing is that the material which you bring to the lab from outside, it should get rid of the soil. So continuous water wash is done with the help of a surfactant in it or any detergent in it. For example, Savlon is used, sometimes you can use any detergent. Then subsequent washing, so that the detergent is lost from the surface. Now then you treat it with a wetting agent. Generally used wetting agents is up to 70% of ethanol volume by volume. This is what is used in plant tissue culture labs. So when I say wetting agents, like for example this 70% ethanol is used as wetting agent. What does it do? From the word itself you can make out wetting agent.
It just increase the time period for the detergent or any disinfectant we are using, just increasing the by making it wet for the longer time. Because if we use 100% ethanol, then it can evaporate easily as compared to 70% ethanol.
Then why at all to use ethanol?
It also
closer to what she said, wetting agent is not to wet, but wet in a sense, and that it would facilitate the penetration of the disinfectant inside the explant, so as to increase the probability of removing all the contaminants or killing the contaminants. Now when I say it increases the probability or the penetration of the disinfectant, how do you think it is increasing the penetration of the
increasing the force
surface related to surface tension? Very good. But what does it do to surface tension? And of what? The liquid in which the wetting agent is there. So suppose you then provide the disinfectant. So what it would do is it reduces the surface tension of the liquid. When you say reduces the surface tension of the liquid, which means the attraction between the molecules of the liquid, so that they can get dispersed and get inside the gaps of the available on the explant surface, and thereby increasing the probability of penetration and disinfection. Yeah, one more point. Now, when you are using seeds as explants to generate either in vitro seedlings or any forms of cultures, sometimes which we observed in lab also for Viola odorata. So, this is one of the medicinal plants. The seeds were not able to give rise to the plants, despite the fact that whatever was given in literature was being provided as the medium for germination. So, that to avoid seed dormancy or to overcome the seed dormancy, then these seeds should be treated with either dilute acids or even heating treatment is given. So, now why do you think and what does the term seed dormancy means to you? And why do you think heating or dilute acid exposure how is it helping in overcoming the seed dormancy? What it must be doing? And of course, after the treatment with dilute acid, then it has to be water washed properly so that the acid is lost. Is there a role of dormancy? So, till the surroundings, think in nature, till the environmental conditions are conducive for the plant to grow or arise, the metabolic activity, it has to be kept viable at the position it is. So, then the metabolic activity has to be arrested. Now, for that the seed dormancy is used by nature. Now, if it becomes so tough that even when the environment is conducive, the seed is not able to come out. So, generally what is needed is there is a covering around the seed which is called as seed coat. So, when the seed coat becomes very hard such that the penetration by water is very difficult, so then it is difficult to rupture the seed coat for the seedling to come out. So, then the acid treatment or your heat treatment can help in removing this coat, so that it facilitates by the exposure of the environmental conditions conducive and the media conditions, the seed can sprout into the seedling. So, how the explant sterilization is done? This is from a Google book where a simple protocol is given. So, generally what is done is you bring the plant material from the outside, you give a water wash continuous, and then with 2% up to Savlon wash can also be given along with water, and then after that you treat it with distilled water so as to remove the detergent, and then after treatment with distilled water you take it in the laminar hood. Now, the laminar hood, laminar hood is what? Have you used a laugh? Yes. So, what is laugh? Laminar air flow. So, why do we use that? Either there is a horizontal flow or there is a vertical flow. So, how is it maintaining the aseptic conditions? So, how is that positive pressure being created? There is a HEPA filter through which sterile air can be pushed in through the mesh with high pressure, so that there is a positive airflow outside the laugh. So, the sterile environment inside, and you must have heard while working in the laugh, it is always preferred to work behind the flame. You lighten the flame and then it is preferred to work behind the flame or close to the flame. Why? Because around the flame again the sterile environment probability is higher because of the heat generated. So, then when the explants once have the surface contaminants have been removed by water wash, now that before you treat it with the disinfectant, there is a treatment given by the wetting agent for 15 to 30 seconds. So, and then after treating it with the wetting agent, again the ethanol is removed by water wash, intermittent water washes is given for every treatment, and then after that you treat it with disinfectants. Now, disinfectants generally used in plant tissue culture technology is either you use sodium hypochlorite, which is up to 20%, or you use your mercury chloride 0.01 to 0.1%. Now, mercury chloride or sodium hypochlorite are your disinfecting agent. So, the exposure time, the concentration of the disinfectant has to be optimized depending on the kind of species, or whether you are working with a tree species, whether you are working with an old explant, whether you are working with a young explant, all that would have to be optimized. So, if you are generally for in vitro cultures, young explants are preferred. Why do you think young explants are preferred? Young explants means parts of the plant which are younger, meristematic regions or younger plant parts. So, why do you think younger plant parts are preferred? I can agree with the meristem, but why do you think then, another question, why less contamination? Very nice, very good. So, because the cells are still rapidly dividing and have not differentiated, the organogenesis and especially the xylem and the phloem bundles have not, which become the throughout connections in the whole plant. So, when it is not there, so the chances of the contaminant, which can be a virus or a bacteria living inside the plant, to get transferred is less. So, that is one of the reasons, meristematic. And why younger plants? Because as you people said, younger plants, the cells are still rapidly dividing. It is still a growing region of the plant, not yet matured. So, because of the rapid division, the ability of differentiating, redifferentiating is much higher than a matured plant part. So, now what is callus induction? Generally in nature, callus is a wound response. Whenever there is a wound created, it is a repair mechanism in nature. So, suppose there is a lesion which is made in the plant. Now, the cells near that damaged region, they start rapidly dividing till they cover that damaged region. And once they cover, why do it? They need to cover it because now the inside of the plant has been exposed. So, it has to be now again protected from the outside environment, which has so many other microbes and maybe pathogenic environment. So, then the cells keep on rapidly dividing which are around that lesion till that portion gets covered, and then once that covering has happened, further on top of it, it is covered by deposition of lignin, wax. Suberin is one form of wax which gets secreted by these cells to create a hard coat or a layer. So, that is how callus is formed in nature as a wound response. Now, when you need to exploit this in vitro cultures, what would you like to have? You would like to have a continuous repair mechanism rather than closing it at any time. So, you would like these cells which have now started rapidly dividing to continue to divide forever till you subculture it and provide medium. Generally, you may see that even small, when you take leaf explants and if you provide the right kind of medium. So, the question was, what do you think will become critical in that right kind of medium to reprogram the cells or to bring them to dedifferentiated states? It is your hormones. So, generally auxins and cytokinins, we will come to that. So, if you can give the right atmosphere and even the environmental conditions, not all species will give a callus induction response if you provide it light conditions. Some callus induction responses from some plants also happen when you put them under complete dark conditions. So, all the conditions have to be optimized depending on species, depending on age of the explant, depending on type of the explant and the genotype. So, how is it induced in vitro? The cells from any plant species can be cultured aseptically in a nutrient medium. Now, any plant part tissue with living cells can be used as an explant, as I said. Then under the influence of plant growth regulators, generally when we say plant hormones, they are endogenously produced plant growth compounds or regulators. But if they are synthetically produced or exogenously provided, then we call them as growth regulators. So what happens when there is an excision and there is rapid division, it will force, because the cells will continue to divide around that region, they will, because you are using a tissue which is an organized culture. So the epidermis, it is a tissue, so there will be a dermal tissue also in that, it is a complete organ which you are using. So once the cells continue to divide, they will rupture the epidermis, the topmost dermal tissue, and the cells will come out, and after few days you will see that there is a blob of tissue which looks like a callus. Now generally it takes 1 to 5 weeks for callus, proper complete callus induction to take place. If you see that even after 2 months or so, or you are not observing complete callus induction or it has become impaired, then which means that either nutrient conditions or growth regulators have to be relooked, and then you should revise the media. Now there can be different forms of callus depending on the species on which you are working. Now this hardness or friability of the callus depends on the cell to cell contact, how closely they are connected with each other, how strongly, and the water content in the callus. So which means the fresh weight to dry weight ratio of the callus. So generally for suspension cultures or in vitro cultures, friable callus is preferred. Friable callus, which means the cells of that callus should be able to get dispersed easily when given suitable conditions, because hard and compact callus cannot be, will become limited wear. What limitation do you think you can face with plant cell cultivations if your beginning material, which is the callus, is very hard and compact?
If you want to exploit plant cells in vitro, you need to mass produce it, isn't it? Now, mass producing the callus, you can do 2 ways. You continue to divide the callus in this solidified medium. Now, you disperse the cells and bring them in suspension, and then continue to divide and scale it up. So, but what is the problem if I continue to do it in the solidified medium?
There the entire tissue is not exposed to the medium. So how does the callus afford to multiply or grow? It is because of that compactness on the solid medium. The cells which are in touch with the medium, they get the nutrients, and then from them it is subsequently transferred to the other cells which are not very close to the media. Now this is a demerit in one sense. What demerit? This also gives heterogeneity to the callus nature, because there are physical gradients, nutrient gradients, so thereby leading to biochemical gradients and maybe their biosynthetic capacity. So that is why callus can be heterogeneous in nature. Now if you need a synchronous culture and all cells giving you reproducible results in terms of productivities, then all cells have to be given the same environment. So if the cells get dispersed in the liquid medium, all cells are getting exposed to the same environment. So the chances are high that the culture would be synchronous in nature, and then the mass transfer limitations which you people were referring to will also be overcome to some extent. Now because they are on the solidified medium, the callus growth in comparison to cells in suspension is also slower. While if you compare it with bacterial cells, the doubling time is still 24 to 72 hours here, and there in bacterial cells is between 10 to 12 hours or 24 hours. Now callus inductions, seeds are preferred as they can withstand severe sterilization conditions. So I would still say it depends on the kind of culture you would like to, even if you see the books, different books say different things, but it is very species specific. So generally if you want to end up, depending on the objective of your study, if the objective is to produce a particular phytochemical from that cell using plant cell cultivation, then in the end what do you need? You need a very high yielding cell line. So for generating a very high yielding cell line, product yielding cell line, your starting material, if it is a high yielding, the chances are all the cells there are high yielding. So if you can get a callus from that explant which is high yielding, the probability is high that you will end up in plant cell lines which are high yielding. So ideally what should be done is, you should pick up a plant variety which is high yielding. For example, I will give you an example. I worked with azadirachtin production. So there are, neem is ubiquitous in our country. It can be found in many regions of the country, but because of the geographic and climatic variation, even in the neem variety which is present in different parts of the country including North and South India, the yields are variable. So, you must pick up a variety which is higher yielding, average high yields are there, and then you must pick up that explant or that region of the plant which is highest yielding in that plant. So, for example, in azadirachtin, seeds generally have the highest yield of azadirachtin. So, we picked up a plant which is in I think a Trivandrum variety which was very high yielding in comparison to there were plants in IIT Delhi also. But then we made a comparison, we picked up the plant from here in South India which was showing high yield, and from that plant we picked up the seed of that plant because the seed is known to have highest yield, and that seed was used to generate the callus. So, what I mean to say here is it is not necessary that you need to pick up the seed only. You must pick up that explant or part of the plant which shows highest yield of that particular metabolite. So, once you have, suppose the callus, successful callus induction happens after 2 to 5 weeks, then how do you subculture? Let it grow for some time. The initial 2 to 3 subcultures, the entire callus which has been induced should be transferred to the fresh medium. You should not disintegrate it because factors like minimum inoculum density, which you must have heard in your microbial fermentations or fermentation technology, that critical inoculum density is crucial for the growth, subsequent growth of the culture. So, you should not, the minute the callus has come out, that does not mean that you rupture it, disintegrate it into fewer smaller sizes, and any size or any small amount of cells would lead to multiplication. There has to be a critical size, a minimum inoculum size which is needed for multiplication further. So, once it has grown to some 10 millimeter cubes or so, then you take that callus after 3 subcultures of the entire callus blob tissue, then you can divide it into 2 and further subculture it for propagation. Now, coming to the suspension culture. So, now how from suspension culture induced from callus? Once you have got the callus, then you bring it in suspension. There are different ways in which suspension cultures are created. Either you can use Buchner funnels, I will show you a picture. So, either you can use Buchner funnels, and if you want a very synchronous culture or very fine suspension, then you can even pipe it out few cells, else people have done callus induction with single cells even, and then subsequent cell suspension also with single cells or small amount of cells. So, you can pipe it out the cells and then use that as inoculum, or you can directly filter the remove the settled aggregates and use rest of the cells, remove the liquid part and remove rest of the cells as filtrate to be used for suspension culture initiation. Now, what is key here is that as you do in fermentation of microbes, inoculum age, inoculum size has a role to play in your growth and production kinetics. Same is true here. Now, it is not that if you directly take the cells, they will start dividing. It depends on what all conditions, it depends on the production medium. Now, if you suddenly bring the cells from your inoculum preparation medium to production medium which is completely different, the cells may not divide, or there may be a long lag depending on the size of the inoculum which you have used. So, the production media, then the conditions which you are using, in the environmental or the media composition which you are using is critical, then your minimum size of the cells which you are using is critical. Now, there are techniques how you can reach to synchronous cultures. Now, for doing synchronous cultures, people use sieves, cell sieve methods. Now, this can lead to the filtrate, different filtrates which will be obtained from different mesh sizes can then be used for generation of suspension cultures as with a high probability that they would be all synchronous in nature. Now, why do you think these all filtrates, suppose I use 80 mesh size or 150 mesh size and I use that filtrate to generate a cell suspension? Why do you think that this increases the probability of getting a synchronous culture? So, because as she said, with the same size of the cells coming in, same shape of the cells coming in the probability is high that their metabolic activity or their age would also be same. So, thereby leading to synchronous cultures. And when I say nurse tissue culture, there is a nurse tissue, nursing tissue involved. So, in this technique what is done is you take a callus, small piece of a callus of the same variety or of a different variety and put it on a sterile environment on a petri dish and put a small sterile filter paper on top of it, and the metabolites or the liquid which gets released by this fresh callus which is nursing will wet the tissue on top of it. Now, this wet tissue which is now supposedly having all the nutrients and other metabolites which have been released onto that tissue, you then inoculate your single cell using the pipette. That single cell from which you need to generate the callus. Once it gets the nursing from this parent callus tissue underneath the filter paper, which is wet filter paper, you will see that the cells will begin to divide. The cells, as they divide, after some amount of division, you will find the callus become visible, and then use that callus for subsequent subculturing can lead to a real synchronous callus or cell culture, because you have started with single cell leading to its subsequent clones. So, this is what is called as single cell culture technique, and this is to achieve synchronous culture, what I was talking as the cell sieving method. Now, another way of carrying out this single cell culture technique is also microculture chamber method, which used to be used, which can be used even now, in which you use a sterile petri dish. That sterile petri dish, you put paraffin oil on top of that sterile petri dish, and then you make use of sterile cover slips. I hope you people know what are cover slips. Now, these take 3 cover slips. 2 cover slips, when they are put on top of that glass slide, right, with the oil, they will form this gap which is shown here, this gap between the 2 cover slides. Now, because there is oil underneath, it will form a small oil boundary, so as to give a sterile environment, so that whatever is inside now in this square would not be in touch with the outside world. Then you inoculate your 1 drop of liquid nutrient with your cell inside that gap, and then you cover this with the 3rd cover slip on top with the paraffin oil. So, now it becomes like a square small chamber, mini chamber or micro chamber, where that single cell is in the drop of the liquid nutrient, and then you can see under the microscope, let it divide. The cells would divide, and then once you have seen it has reached to some groups of cells mass, then that can be subsequently transferred and can lead to generation even of the whole plant. So, this is called as microculture chamber technique. So, before we begin with different forms of culture, it is important for us to know 2 things. One is, how will we find whether the cells are growing, and the second is, how will you find whether the cells are viable. So, how do we find, what parameters do we use to quantify the plant cell growth or plant tissue growth? There are different ways. One is, you do simple dry cell weight estimation, in which you dry the biomass in oven at 60 degrees C, the plant biomass or the callus or the tissue biomass, and let it dry until constant weight is achieved. So, that is simple, you calculate the biomass concentration. Now, the second is, you use packed cell volume, because plant cells are large in size, this works appreciably well. So, you can use a centrifuge tube and then let the plant biomass, because they form aggregates, they settle, you will see that the plant biomass after some time will settle down, and then in comparison to the total volume occupied in that centrifuge, that percentage which the biomass occupies is represented as packed cell volume. That can also be an indication, a rough indication for growth of the biomass. Now, what else? Fresh weight to dry weight, which is called as growth index. Now, growth index is another parameter which is used when you measure the growth of the callus, which is final fresh weight after some amount of time. You calculate the fresh weight final minus the initial fresh weight, which means the amount of fresh weight which has increased to the initial amount of fresh weight which was there. So, this ratio is called as growth index and is a parameter which gives you an indication of how much growth has happened. Now, talking about plant cell viability, there are many different ways, but here I have listed down 3 different ways which are generally used in literature to determine plant cell viability. One is the fluorescein diacetate vital strain. So, this particular strain it is based on the activity of esterases in the viable cells. Now, these enzymes can reduce this dye to produce a fluorescent product which you can then, because the cells are viable and the membrane integrity is in place, so they can hold on to the fluorescent product which you can see under the UV light, and percentage viability is given as the number of fluorescent cells to the total number of cells. So, those which are fluorescent would be viable because it shows integrity of the cell membrane still in place, and it also shows the activity of esterases in the esterases are still active, metabolically active cells in the viable cells. So, this is how percentage viability can be calculated. The second way is using tetrazolium salts, which you must have heard even in your animal cell cultures and others, MTT assay. So, TTC is a triphenyl tetrazolium salt. It is used and it generally is dependent on the activity of dehydrogenases enzymes. Now, the dehydrogenases it signifies the metabolic activity of the cell, because whether the mitochondria is actively functional or not. So what it does is, because it is dependent on dehydrogenases, so once it reacts with this tetrazolium salt, it forms a red formazine product. This red formazine product can be measured spectrophotometrically, which can then be used to calculate percentage viability of the cell. Now, Evans blue, any kind of such stains, they depend on whether, now once the cell membrane the cell is alive, the membrane integrity is in place. If the membrane integrity is in place, it is selectively permeable. Now, selectively permeable, either you can use not taking up a particular strain as a viable marker, or taking up a particular strain and not letting it go as a viable marker. So, in Evans blue, dead cells become blue, which means what are you looking at? Because the cell membrane integrity of the non-viable cells is not there, so the dye will be able to get in and stain the cell. So, that is why Evans blue is can be used for determining the viability of the plant cells. Applications. So, in agriculture, in what way? Production of higher yielding or drought resistant or herbicide resistant or salt resistant species of crops. Then horticulture and forestry, if you want to do micropropagation or if you want to produce synthetic seeds. Then in production of phytopharmaceuticals, your callus cell suspension cultures or hairy root cultures, which is organ culture, comes to play a role. Then products from transgenic plants, you must have heard about edible plants or GMOs, or plants with specific nutrient quality like your golden rice, or plants with which can be protected from certain insect pests. So, for example, your BT cotton. Okay.